So welcome to this session of STEM Sparks. I'm excited to have José Zepeda, who's going to talk about his work in neuropharmacology and how he got there and what he's involved in now. So thank you, Jose, and you can feel free to share your screen.
Thanks for having me. All right. So can you see that all right?
Yep.
Okay, perfect. So hello, everybody. Thank you so much for taking time out of your day to come listen to my talk. My name is José Zepeda. I'm a second year pharmacology student at the Vanderbilt University School of Medicine. And today I'm going to be talking to you about neuroplasticity. But before I go into that, I'd really like to start with telling you who I am. So I was born in Yuma, Arizona. I'm the son of Mexican immigrants. And at around the age of 2 or 3, my family moved up to Boston, Massachusetts, or Cambridge, Massachusetts, to be more specific. I lived there up until I was 12, when something very unfortunate happened, which was that my mother was actually deported to Mexico. So my sister and I followed my mother into Mexico, where I would then reside for about 7 years. And so while there, I attended school in Mexico, and I worked all sorts of jobs, primarily in agriculture. So I lived in a border town, which is just south of Arizona. So it would be, let me see if I can get my pointer going here. So it would be right here in this sort of armpit of Mexico, where there's the border between Sonora and Arizona. As I mentioned, I was working in agriculture, and so as many of you might suspect, agriculture does not pay very well. It's also a very grueling job. And so I got quite fed up with it and the few economic opportunities that there were, and decided to save up some money and move back to Boston. So I moved back to Boston at the age of 19, where I then finished my high school at Chelsea High School in Massachusetts. After high school, I decided to pursue biochemistry. So I attended the University of Massachusetts in Boston, where I received my bachelor's in biochemistry. And now, sort of breaking this sort of jumping between the coasts, I'm now living in Tennessee. As I mentioned, I'm studying pharmacology at Vanderbilt University, and I'm specifically studying within the field of addiction neuroscience. So stepping back a bit, as I mentioned, I went to Chelsea High School, and while attending Chelsea High School, I was absolutely certain that I was going to be a physicist. I took my first advanced physics course. I was reading a lot of books on physics, and I was just sure that this was going to become my career path. But the same thing that sort of drew me into physics kind of took me away from physics. I was also reading a lot on philosophy, and I came across this book which would forever change my life. It's titled The Outer Limits of Reason by Noson Yanofsky, and it focuses on philosophical conundrums, which simply put are sort of these deep questions that lie at sort of the edges of our knowledge, many questions which we believe might not even have an answer. And so while reading through this book, I learned about double pendulums. So double pendulums are this really interesting system. They're very much like your usual pendulum, which, can just swing back and forth, except what you do now is you attach another pendulum to that first pendulum to create the double pendulum. And from this very simple system arise this very elegant chaos or dynamicism from this system. And so I found that really cool to read about. And then I eventually read within the same book even that it was impossible to repeat the swing of a double pendulum twice. Meaning that if I lifted this double pendulum and I let it go and I tried to raised that double pendulum to the same point, I would not be able to replicate its swing perfectly. I would need an infinite amount of precision to do so. And so a picture is worth 1,000 words and a video is worth 1,000,000. So I'm going to try to play this here. Let's see if this works. Okay. Can you see that fine? So this here's a double pendulum. And what you can see is that after I lift it to a certain height and then I release it, you can see that the double pendulum follows this very erratic behavior. So just play that once again. And so this trajectory very quickly becomes impossible to predict within our physical world, at least. And so these chaotic trajectories are governed by these very elegant mathematical equations. And so although I don't have time to go into them, if you're interested at all in these systems, there's some online simulations that you can find if you just type in double pendulum chaotic simulation. And you can play around with all of these parameters and see these swings for yourself and mess around with them. Now, in that science fair project, my partner and I, Robert Tempelhoff, showed that you could change the level of chaos within the system by changing the amount of energy that you input into the system. So very simply, if you raised the pendulum higher, you would have a higher level of chaos and so forth. And there was a limit for this, and I won't go into that. But it was really interesting to see that how this system could be manipulated and the predictions that we could make. We also showed that at different temperatures, even from the same angle, you could get these vastly different levels of chaos, where here at 30 degrees in Fahrenheit, you can see that there's a lot more spins. It becomes a lot more difficult to predict. And I'm not going to go into that for the sake of time, into how we define chaos, but essentially it's not too far from what you would imagine of just being unpredictable. And so while at this science fair, I underwent a scenario that was very similar to what's going on in this picture. I was presenting my science fair poster to all the judges and people who were walking by when I was approached by a professor at MIT. And so this professor mentioned to me that they had actually done their PhD modeling the brain as a dynamic system. And so this really got me going. I had been reading a little bit about the brain, but I'd never really thought about how complex it actually was. So being one of the things that attracted me to physics was actually the complexity of our natural world. And so to think about the brain and how it actually is one of the most complex, if not the most complex system in the world, really steered me in a different direction. And so this is sort of what the brain organized as a dynamical system might look like, where you might have one neuron that will fire, and that neuron is attached or communicating to about 10,000 other neurons. And so before you know it, if one neuron fires, it might be causing these other 10,000 neurons to fire. But of course, this very quickly becomes complex, where the initial conditions of the system could lead to these vastly different outcomes. And so I began to think of the brain as sort of this orchestra of double pendulums, and I became increasingly fascinated by it. And so when it was time for me to decide what I wanted to study for in college, it was very easy for me to pick biochemistry, which sort of lies at the foundation of the biochemical organization of the brain. Now, I didn't go into studying the brain as a dynamical system or even computationally, but instead I've been looking at something a little bit more fundamental, something termed neuroplasticity. So neuroplasticity simply refers to the brain's ability to make, break, strengthen, and weaken neural connections. And so there are various different mechanisms that govern this process. that will strengthen or break these connections. And we know that neuroplasticity is fundamental to learning and memory, and many diseases can arise when neuroplasticity goes awry. And so during my undergraduate career, I had the amazing opportunity of being able to study synapses at MIT. And so here I want to show you some of the imaging that we were able to accomplish. Let's see if this works. Okay. Is that a still image? Okay. Try that again. Okay, now is that working?
Yep, that's working.
Great. So what you can see now are that these synapses are lighting up green. And so what we've done is we've injected a single neuron with a tool known as GCAM. So GCAM's a calcium indicator, and it allows us to visualize increases in calcium at these synapses, which are essentially a proxy for neural activity. So we're able to look at these synapses and see when they're becoming active per se, and we can use this strategy to identify the functional identity of a synapse. And so this would be important for then making predictions about whether this synapse might be strengthened or weakened, given some behavioral experience or so forth. But of course, once we've determined that, the next step is to determine what the molecular mechanisms behind this plasticity might be. And so one other strategy that we've used is known as mass analysis of the proteome, or expansion microscopy, where we can essentially grab a brain and expand it linearly so that then we can zoom in at these single synapses and look at their protein content. So these are unlabeled because this is currently unpublished data, but what you can appreciate is that we're able to visualize protein content at single synapses with extremely high resolution and precision. And so using strategies such as these, we've been able to uncover some fundamental rules about neuroplasticity. And so here I'm highlighting this rule known as compensation, where if a certain synapse is strengthened, this will cause neighboring synapses to decrease in size. And this makes logical sense when you start to think about what might happen if all of the synapses on a neuron were strengthened. It could overwhelm a neuron, and this could lead to toxicity that would then lead to the neuron's death. So this is essentially a neuron's way of controlling the amount of information that's flowing through it. And so you might be wondering, well, why is synaptic plasticity so important to me? Well, I hope I've been able to convince you that synaptic plasticity is a rich area for discovery and that it's really important for various brain processes. But as I mentioned earlier, I grew up in a border town where, unfortunately, there's a lot of illegal activity regarding the smuggling of illegal addictive substances such as fentanyl, opioids, and methamphetamines. And so there's drugs being crossed through the border illegally every single day. And when you live in a border town, unfortunately, what this leads to is a lot of violence and a lot of involvement by people who live in the border town in these smuggling activities. Another consequence is that there are very high rates of addiction in these communities. And unfortunately, some of my family and friends have been affected by substance use disorders. Now, substance use disorders are not confined to border towns. We can see or appreciate from this visualization here that substance use disorders are occurring all over the nation, and that there has been an increase in the use of opioids and opioid overdose rates, and there's really been no show of decline. So this means, and currently, there aren't really any good therapeutics for substance use disorders on the market. So this begs that we study mechanisms that might be underlying substance use disorders to develop new therapeutics. So the area of the brain that I focus on is known as the nucleus accumbens. So the nucleus accumbens it forms part of what is termed the reward circuitry of the brain. So this reward circuitry of the brain is in charge of the motivation that you might feel to walk to the corner store to buy some food, as well as so many other motivated behaviors. But what one can imagine is that in the case of a substance use disorder, this wiring goes awry, and these communications become very strengthened. And specifically within the nucleus accumbens, I'm interested in a group of neurons known as calretinin interneurons. So an interneuron simply means that they cast information locally and not to other parts of the brain, but are able to modulate principal neurons, which do send connections to other parts of the brain. So you can imagine that interneurons are able to highly control how information propagates through this brain region. One such possibility for how the calretinin interneurons might be transforming information traveling through the area is through a circuit motif known as feed-forward inhibition. So feed-forward inhibition refers to this scenario where there's excitatory input, meaning that this communication increases the chances of a neuron firing, landing onto an interneuron, which is inhibitory and actually decreases the chances of this principal neuron firing. So what we can imagine might happen here is that this information, this excitatory drive, enters the system. It might cause this neuron to fire and output information, but very quickly inhibition is recruited to silence this neuron and prevent it from communicating further. So these interneurons are able to highly modulate how information travels through this region. And I believe that calretinin interneurons might be embedded in this kind of network. And so one way of determining whether calretinin interneurons might be partaking in feed-forward inhibition is using a technique known as whole cell voltage clamp electrophysiology. So using an ultra-thin pipette, I can rupture the membrane of a neuron so that I can then use an electrode to record different currents, electrical currents that are traveling through this neuron. And so we know that electrical currents are, in a sense, the language of neurons. Neurons are able to open ion channels and allow the influx of ions that will then cause these electrical charges across the neuron's membrane. And so we can record these. And using this, we can figure out two things. One, how is it that these calretinin interneurons are embedded within the nucleus accumbens? What do they communicate to? What do they receive communications from? As well as what kinds of synaptic plasticity these interneurons might undergo after, say, a mouse experiences an addictive substance such as cocaine. And so I hope that you've been able to appreciate how vast and expansive neuroscience really is. It's a very interdisciplinary field, and understanding the brain, so complex as it is going to require the collaboration amongst so many different subfields within neuroscience. And very briefly, I do want to say that if you were to pursue something like neuroscience in college, one possibility might be to become a professor one day and teach neuroscience, as well as to have your own lab. Or you can go into industry where you can also be a scientist working for a biotechnology company or a pharmaceutical company. There's also areas of neuroscience and its implications for law and policy, among so many other possibilities. So lastly, I want to thank my lab, the Gruder Lab at Vanderbilt, and my mentor, Brad Gruder, as well as my lab mates, as well as my previous lab at MIT, the SIR Lab. And this here, these names, Jacques Ip, Katya Zimring and Kyle Jenks. Together, we make up the synaptic plasticity team at MIT. We still meet every other week to talk about ongoing projects and so forth. And I'm extremely grateful for their mentorship, as well as my funding resources from the National Institutes of Health and the National Science Foundation. And with that, I thank you for your time, and I'm happy to take any questions you might have.
Awesome. Thank you, Jose. So for those who are joining in, feel free to put a message in a chat or raise your hand. If you want to close out your slides, that would be cool. So I think it would be helpful. I'd love to hear a little bit more about like what your day-to-day looks like. Like how much time are you spending in the lab? What kind of schedule do you have? That sort of thing.
Yeah. So as a graduate student, it highly varies. One really nice thing is that lab work is highly flexible. You can sort of make your own schedule. Now, this is going to vary from lab to lab, but if you have a flexible PI like I do, who doesn't really care when you get the work done, as long as you get the work done, then, you can really, plan around significant family events or things that you need to do. And also there's a lot of flexibility around coursework your first 2 years. So you will be taking, didactic coursework such as you do in undergrad, and probably coming into lab a lot less when you have exams and so forth. So I try to be in lab as much as I need to, and not too much more than that.
So how many brains do you think you dissect in a given week or month?
Yeah, that could be, it really is going to depend. There's some weeks where I'm dissecting 3 brains a day, or there are weeks where I don't dissect a single brain. So really it depends on what experiments I have planned for that week. I probably have dissected a lot of brains at this point though.
Yeah. There's a question from Camilla. What tools and machines do you use for your research?
Yeah, so that's a great question. So I showed you some of the imaging earlier, and so the in vivo calcium fluctuations that I was showing you, those were recorded by a system known as a 2-photon microscope. It's this really fascinating microscope, which also has some really cool physics properties behind it, where essentially it allows you to image live brain tissue in an alive and awake mouse without really damaging its brain, because the excitation is minimal. We also use other microscopes such as a confocal microscope, which allows us that super high resolution that I was showing you, that allows us to look at protein at single spines. And then lastly, the recording electrode and glass pipette setup is performed on an electrophysiology rig. And so this is a specialized rig that allows us to keep brain slices alive and the neurons within them alive, so we can go in and essentially rupture the membranes and record from them. Also has a microscope attached to that, because you wouldn't be able to see a neuron without a pretty powerful microscope.
I think one thing too that's so important with that too, and this came up in the question earlier, and it just kind of struck me in terms of in the earlier panel presentation around having access as a high school student to labs and to equipment. And I think that one of the pieces that's so important about the Science and Engineering Fair project system is that you get to learn all the pieces and projects of doing real scientific research, so that when you are in undergrad or graduate processes, you've got a lot of those skills down. So then when you do have access to this highly technical equipment, you've got a leg up in terms of knowing the other stuff you need to do, that you can really focus on using machines. Do you feel like that sort of is reflected in your experience, Jose?
Absolutely. Yeah. I mean, I think one of the most important things in science is being able to develop hypothesis-driven questions. So having some possibilities, interesting possibilities in mind when you're coming up with these questions, But probably more important than that is having good controls. So making sure that when you have finished your experiment, you can make a decisive statement about your findings, and leave as many little weaknesses in your experiments as possible. So I think that I got my firsthand experience of that at the science fair, and that's definitely something that's helped me throughout the rest of my scientific career.
Great. So where do you think there are some trends in your field in terms of areas where it could be helpful for students to look into, or if there's data out there or information to splice and dice?
Yeah, I think I mean neuroscience is a pretty infant field. It hasn't existed for much more, if more than 100 years. And what I was going to say is that right now, and this has probably been a trend in the science fair, is the power of computational methods. And so the brain being such a powerful computer, we do require people with computational backgrounds to try to understand exactly how information is being processed. So I think that the field of computational neuroscience is growing very quickly, but also computational drug discovery, which is a relatively new field across so many different biomedical systems. I think that there's so much unlocked potential in um, computational drug discovery methods, where you play around with these different protein models, and you simulate drugs attaching to them to sort of give you an idea of what might work, and it could save us a lot of money and make us more efficient. So I think that those are two areas that have extreme potential within neuroscience right now.
That's great. Yeah, and I'll put a plug in the next session too. One of the next sessions is around neuromarketing and understanding the sort of the brain's response to pleasure and those pieces as well, and sort of how that plays in. So it's all about the neuro, right?
Yeah, you can essentially attach neuro to just about any scientific
Yep.
field, and there'll be some niche field on it. Neurochemistry, neuromarketing, you name
Yep.
it.
Yep. Okay. Well, if there aren't any other questions, I want to thank you so much. I think this is such an interesting area. I think your story is so great and inspirational that we really appreciate the time that you've taken.
Yeah, thank you so much for listening.
Great. Awesome. Oh, thank you.
Thank you.